is the National Research Council Institute for Marine Biotechnology and Research Management. So he is the director of the institute that has several locations in Italy. How many locations you have here in Italy?
4.
4. How many people working for your institute, more or less?
120 fixed position, and something like 40 or 50 young researchers and PhD, including PhD as well. Yeah.
Okay, so that's one of the things that Gianmarco is running, but he's also 2 years ago in charge of running one of the main project that the country is actually running, which is that you know very well, is the National Recovery and Resilience Plan. You heard PNRR, this word many times in all the newspaper. Gianmarco is together with the National Research Council is coordinating the National Biodiversity Center. Within the PNRR, being founded several centers in Italy, several research centers. One is on biodiversity, management and conservation. Among of the National Biodiversity Center, there are work packages dedicated to the marine biodiversity, and Gianmarco is a coordinator of one of these work package that we call Spoke number 2. So, of course, he's active in our PhD program as a teacher. So I'm very glad that Gianmarco is within all this framework of activity here today with us, talking about his research skills, which is microbiology in general, and now it's applied to fishery management. So thanks again, Gianmarco, and good luck for your lesson. I'm gonna sit there until the first phone call, okay. As you know very well.
Don't worry, and thank you so much for the introduction. So should I speak in English or Italian?
You can, it's better, yeah.
English, okay. And yeah, I'm really happy to be here because the role of director that Stefano was telling a few minutes ago is taking me quite far away from occasion like this to be teaching and to be close to my research line. So I'm really happy to be here and spend some hours with you. And I really will appreciate if the lesson will be interactive. So feel free to interrupt me and don't be afraid. I don't speak slow because we have many hours. I have about 100 slides, but yeah, don't worry, interrupt me at any time. Okay, so this work. Okay. So what I'm going to talk to you is basically to describe something that is really related to my job as a scientist. So I've been working in marine microbiology in the last 22 years, I'm getting old. And so the lesson will be, according to the title, specifically to the microbiome of fishes, but I want to spend with you also some more general slides on the role of microbes in marine ecosystems, because I think it is really important that everybody of you has really understood why microbes are so important for the functioning of our seas and ocean. And to do this, it is really imperative to go through the history of modern techniques that we now use very commonly in our laboratory, but 20 years ago when I started to do this were really very far even from being imagined, because technology, particularly DNA technology, is moving so fast, has moved so fast in the last decades. And so I just want to go with you across all the different steps of the last 20 years, because I think it's also important for the young researcher to understand how things change very quickly in marine science. And then I will speak about the teleost microbiome, and particularly with the research that I'm carrying, particularly with the help of some of you, particularly Marco and Naomi. And lastly, a few slides on the safety of fish and fisheries products. This is a slide that we typically start with every time we have to speak about the marine microbiome, and it is a very important paper that came out in 1998, and it was during my, I was still a student, but it was really an important paper that came out on PNAS, and it is very famous because while it was known in the previous years, it was something that was emerging from the 70s, that actually marine microbes were really important for the functioning of marine ecosystems. This paper was the first one that showed to all the community of scientists, not only to the specialists like we were at that time, that prokaryotes are really the unseen majority of life on the planet. So it is a very simple title, and this is important also for you, for your career, when you speak about your papers, and when deciding the titles, look at this title. It is very clear, very simple, but had a huge impact on the community of not only of marine scientists. And what these people did was to simply take the literature and make calculations and show to the community that actually the amount, the number, so in terms of density, the amount in terms of mass of prokaryotes is so significant on Earth that should not be neglected in the future studies. And so they make very precise calculations, and particularly on the number of petagrams of carbon that is made up of all microbes. So this is not a marine specialistic paper, but they were making calculation seas, oceans, earth, air, and also the subsurface. That was a really emerging topic at that time. So all the study of microbes that lives below the surface of sediment, for instance. So what was called at that time the deep marine biosphere, which was important for that paper because that type of environment is dominated by microbes. And so if we go back to the marine ecosystems, it will be very easy calculation for you if you just think that one single drop of seawater, so just a drop, maybe 1 milliliter, contains 1 million of microbes. So it's really a huge number of microbes. Just consider how big are our seas and ocean. And this presence is even larger if we take the same amount of volume in the sediment. So if we take just one single gram of wet sediment, you will find 1 billion on microbes in each gram of sediment. And so these are some examples also that we use just to show, to make easy comparisons. For instance, we say that when there are 2 multiplied 10 to 29 bacteria in the oceans, and this number is lower than the number of stars in the universe. And also, if we take at viruses, so that people tend to call microbes as well, but we know that viruses, they don't have a cellular organization, so they are acellular agents. But typically in microbial ecology, they are considered as microbes as well, even if the term is not really proper. So if we could take all the ocean viruses along in a single viral string of pairs, it would belong 400,000 light years. So it's really a lot of microbes. And if we take all together and we try to calculate as Whitman did, the cumulative biomass of marine microbes is equivalent to 50 million blue whales. The blue whales are the largest organisms that populate our seas and oceans. And also we have to consider that we have 300 petagrams of microbes in the ocean subsurface. So moving from below the first surface of the seafloor for hundreds and hundreds of meters, we also have microbes, and these microbes taken together add 300 petagrams of carbon. So it's really a huge number of microbes. They are not only abundant, but they are only extremely biodiverse. So biodiversity of microbes is really enormous. Here you see just a number of different shapes and forms, but we will learn in this lesson that morphology is not really informative for marine microbes. So if you can see here, and this is particularly true for microeukaryotes, that morphology can be important for taxonomy. This will not be the case for bacteria and archaea, because they have very simple morphologies. So they are cocci or bacilli or vibrio, but there may be hundreds of species having the same morphology. But this is just an example of the different morphology that we have. And there were very interesting papers coming out in Nature and Science at that time. So I think it was something like 15 years ago. And yeah, the one that you see here is taken from Nature. So moving to their role, we have to consider that their role is so essential for the functioning of the ocean. And this is because of a number of reasons that I will try to explain here. One of the simplest is that they are really the first inhabitants of our seas and oceans. So life began in our seas and oceans a long time ago, 300.5 billion years ago, and microbes were the only life form for many, many years. And so they are believed to have created the condition for the higher life forms to develop in subsequent years. So they are really important because they created that conditions that allowed marine life to grow later on. And as a marine microbiologist, sometimes it is really common for me to understand how people is really not aware of the role of microbes in the ocean. And I have this that is a very simple video that I will try to run. Okay.
This is a phytoplankton. These tiny algae harvest energy from the sun to grow and reproduce.
Let me see if I can share. Try here.
Hai condiviso lo schermo, vero?
Sì.
Allora dovrebbe... Copia e incolla la stringa nel browser.
Proviamo,
Secondo me dopo funziona.
Succede nulla. Eccola. Adesso? Ok.
This is a phytoplankton. These tiny algae harvest energy from the sun to grow and reproduce, providing food for the entire ocean food chain as one organism feeds on another. This was how scientists traditionally occasionally view food webs, small things being eaten by big things. But this view leaves out one piece of the food web puzzle. Zoom in to a size 100 times smaller than the width of a hair, and you'll find the invisible missing link, a microbe. Microbes are easy to miss, and even scientists only relatively recently discovered just how abundant they are in the ocean. There are millions of bacteria and other single-celled organisms in just a spoonful of water. Microbes have many roles in the ocean food web. Let's explain. Ocean creatures are very sloppy when it comes to eating, so it is up to bacteria to clean up the mess. When tiny animal grazers like zooplankton chew through phytoplankton, they lose much of what they aim to eat. The phytoplankton are ripped apart by munching jaws, and the remnants of their food are released into the water. Microbes can then swoop in and absorb the tiny molecules that would otherwise go to waste. Microbes also get a nutritious feast when a phytoplankton is attacked by a virus. When the host cell dies, it expels its contents, including the viruses, into the ocean for the bacteria to feed on. Other ocean creatures expel nutrients in their excrement. All of this is fair game for hungry microbes that make up the diet of the ocean's tiniest grazers. The discovery of the microbial loop of dissolved molecules to microbes to grazers revolutionized the way we think about food webs in the ocean. Ultimately, it is thanks to this invisible cleanup crew that our ocean is a clean and clear home, where unbarred life can thrive.
Okay. Let's go back. Okay, so I don't know how many of you were already familiar. Okay. Okay, so this concept is really important for all marine biologists and ecologists because it really explains how microbes are important along the food webs. And if we go through the history of this concept that you can find now on the textbooks of university and so on, it is important to understand who and to remember who the authors of this concept are. And this is the paper, which was authored by a group of scientists a long time ago in 183. You can find here the original paper. And I think it was a really elegant paper at that time that built the concept. So there were an emerging number of evidences from the 70s that were showing that microbes were important, were abundant, had some roles, but this group of scientists, which has now passed in the history of marine science, was able to conceptualize all this concept and to make a picture, to make the picture of the microbial loop, which is important because if we wouldn't have a microbial loop, we wouldn't have any fish in our seas. Because the microbial loop, from the quantitative point of view, is a loop, is a pathway where up to 90% of the organic carbon pathway passes by. So how we can explain the microbial loop? So if you look here, okay, here we go. If you try to look here, this is the typical food web that was called food chain when I was a student at high school. So you have the classical phytoplankton, zooplankton, predatory fish, and apical predators here. And this was what was believed until the microbial loop concept development, the main process of passing of matter and energy in the marine ecosystems. What these people understood and conceptualized was that from every trophic level of this chain or this web, there was a loss. There was a loss of carbon, which was typically in the form of detritus, so dead organic matter, fecal pellet, exudates, dissolved organic carbon, and so on. And all these organic matter that can account for up to 90% of all the organic carbon and matter that is cycled across the classical food chain would have been lost without bacteria, without the degradation by particularly heterotrophic bacteria that can consume this detritus and can transform this dead organic matter into biomass. So they can convert as decomposers something that is dead in something that is living, which is their biomass, and then they are grazed again by other organisms, and so all this detritus is recycled. So by using a term that was not used at the time of Azam, and it is a classical example of circular economy. Okay, so this is a real circular economy process where everything that is lost from here is completely recycled and taken together into the loop. So this is why the microbial loop really revolutionized the marine ecology. But marine bacteria also, they have many other important roles. So that role is specifically used for bacteria that consume, that are able to degrade organic matter. But also we have autotrophs, and particularly bacteria that have the molecular machinery to fix carbon dioxide. And they are particularly relevant, especially in the sunlit ocean, and particularly in the surface ocean, where there are two taxa that belongs to cyanobacteria, and particularly Prochlorococcus and Synepococcus. They are known to produce a lot of oxygen. And something that we used to say typically is that we have to thank marine bacteria for one breath every two that we have, because we have papers that make quantitative estimates of the amount of oxygen that is produced by marine bacteria and by phytoplankton, as opposed to the oxygen that is produced by terrestrial photosynthesizer, and it is typically half and half. So marine microbes produce half of the oxygen that we breathe. And also the role of marine microbes is not only in the oxygen cycling, not only in the carbon dioxide cycling, but is basically in all the most important elements. So it is generally told that marine microbes drive marine biogeochemical cycles, and this is true. This is true because basing on the nearly infinite interactions that marine microbes perform at the very small scale. So we are speaking about millimeter scale, micrometer scale, but this is, if we amplify this for the ocean, the role that they play in the biogeochemical cycle is really relevant. The one that you see here is just one example, and it is particularly related to the biological sequestration of carbon in the sea. This is what it is called the biological pump. And you can see that, for instance, in the cycle, the role of phytoplankton and microbes can sequester carbon and also can transfer through the biological pump, so through the sinking of cells, of organic matter, of particles, of marine snow, so to sequester into the deep ocean interior. And this is something that also is really helpful in this time, particularly thinking about the climate change and all the overproduction of CO2 due to the anthropogenic activity. So I've been speaking about the seawater and the seafloor. So these are the most known and most abundant microbial habitats. But they are not the only habitats, because we can say that every single surface in the ocean is heavily colonized by microbes. There is no microbe-free environment on Earth, and it is the same for the ocean as well. And so if we think to seawater and sediment, so to the seafloor, there is also a growing recognition, and this is also part of my talk, that also the surface of marine organisms is heavily colonized by microbes. So we can use the terms epibionts to refer to the microbes that live on the surface, but of organisms. And when I say organisms, I'm referring to microbes, for instance. So phytoplankton cells, also they have the microbiome, going on to the higher trophic level and also to the fishes, of course. But a term that has been increasingly used now is holobionts. So the sum of microbes and of the organisms that host them. And also the term microbiome that is increasingly used now. When I was a student like you, that term was not existing anymore. We were using the term bacterial communities, microbial communities, but now I have to recognize that the term microbiome is increasingly used in the common language. It is important for you that you understand the differences in terminology, because some people are using microbiome and microbiota as synonymous, but this is not the case, because when you speak about microbiota, you're referring to the cells, to the organisms, to the biota. But when you use the term microbiome, you're referring not only to the organisms, but also to all the genes that they carry on. So the term microbiota, by definition, includes not only the living cells, but also all the genetic inventory that they carry on. So if you go on the literature, there are also you will see that there is an abuse of this term, but I'm using microbiome as well because it is increasingly used in general as a synonymous for microbial communities. While the study of microbes in seawater and in the sediment has been carried out for many years, I would say that the history, the first studies have been carried out in the 60s, in the 70s. The study of microbes living in close association with marine organisms, so I will use the term microbial epibionts here, is more recent. It's more recent, and what I'm showing here is just an example of a paper. This is a paper I contributed to now more than 10 years ago, and it was just an example. The study was arising from the collaboration with zoologists from the university where I was working at that time, the University of Ancona. And particularly what these colleagues from the zoology group were observing was that this organism, so this is an hydroid, Ectopleura crocea, by analyzing the surface and particularly in some steps of their cycle, it was heavily colonized by a lot of microbes, and particularly two morphotypes. So two morphotypes that is here. One was horseshoe-shaped, this one in green, and the other one was more fusiform, like a worm. And that was a time 10 years ago, at the beginning of the use of molecular methods. So the first attempt that we were using, because as I was saying, for a microbiologist, it's not possible to identify the species by just looking at this picture. So this could be, despite these morphologies were quite uncommon for bacteria, but for me and for my colleagues, was really impossible to name the species. We could just hypothesize that there were two different species because of the different morphology. And so we tried to use cultivation approaches, which were unsuccessful. And also we were collecting information from the colleagues, and we could see together with them that these prokaryotes were present all around the epidermidis, were present simultaneously. And so this was stimulating us to try to give a name to this bacteria. And the first question was to understand whether there were microeukaryotes or prokaryotes, because we had evidence in literature that particularly the worm type could be a very small eukaryotes as well. And so what we did was to use transmission electron microscopy that was available, just to look at the infrastructure. So this was a scanning electron microscope, and from here you cannot see what is inside the cell. So you could just see the external morphology. But using TEM, we could see also the ultrastructure of the cells, and it was an indication based on the cellular content of the cell that they were really prokaryotes. And so we decided to try to sequence them, and we were using 16S rDNA NGS sequencing that I will explain much better in the next slide. So for now, just take it as a concept. And what we could find in our library of DNA was that there were a lot of other species that you couldn't see here, or maybe you can see, if you look very close, you can see some bacillus, smaller spheres and whatever, and this is what we could see here. So this is a list of bacterial genera that we could generate from our library. But there was a clear dominance of two bacteria, particularly Polaribacter and Dephthia, and then we could go back to the taxonomy and see that actually the type 1 was a Dephthia, because Dephthia is known to present that type of shape, and the other one was Polaribacter. So we could describe the species. The next step would be to understand the role they play, which was not my case, unfortunately, because then I moved to CNR and I did other kind of research. But that was just an example to show how, at that time, 10 years ago, how was complex just to name bacteria on the surface of microbes. And that was the time where the microbiome revolution was starting. So apologize if this is just in Italian, but at that time, and particularly for the human microbiome world, not for the marine, but for the biomedical world, there was an increasing appreciation that humans are not living alone, but humans are holobionts as well, and every one of us in this room has more than 2 kilos of microbes that colonizes our surface, our internal tissue, our gut, our mucosal, oral mucosal, and so on. And this concept of the superorganisms that was really not known for science because of the methodological limitation that I was mentioning and that I will explain better in the next slides. So this appreciation of the humans as a superorganism was really impacting the community. So this was a cover in Nature. And the fact that the human body is composed by 10 or 100 times less number of cells compared to the numbers of microbes that we have. So to make it simple, in our body we have a number of bacteria that we carry on with us every day that is 10 to 100 times higher than the number of cells that make up our body. Of course, these cells are smaller, so in terms of mass, we are more human than microbial, but in terms of numbers and also in terms of genes. So I remember there was another cover in Nature that was called our second genomes, because if we take together all our microbes, and if we take together all the genes that these microbes carry, and this was done in that years by the human microbiologists, we can say that our cell contain 30,000 genes, our cells, but the microbiome that we carry is much more. It's hundreds of thousands of genes. So they really help us to live. And yeah, this is just some dissemination, and it is something that is now more than 10 years ago. And there was a TED that time that is probably still available, that was really impacting, and it was even if it is a little bit aged now. And particularly this guy, which is named Rob Knight, at that time was the one that was using this approach to explore the microbiome. And why this revolution was starting? Because, as I was mentioning, if you want to study the diversity of higher organisms, such as the ones that we see here, I don't want to say that it's easier, of course, it's complex as well, but if you want to describe the species, you can use morphology. Okay, you can really use morphology to understand the difference between a shark and a bird. But if you take microbes, and particularly prokaryotes, this is a very famous picture that every time we use. So it's just a drop of seawater put on the electron microscope, and see, you can see how diverse is the morphology. It's not that diverse because you can see different shapes, but for us it's really difficult to understand what kind of species is this one or this one. You can see that this can be a vibrio, for instance, but there are plenty of species of vibrio. So it is really difficult to understand. So morphology is not or only poorly informative. And so how microbiologists were discriminating, were identifying the species. Typically you have to go in the lab, in the field, collect samples. This is a picture of me working in Australia. Then you had to go back to the lab, take that sample and prepare different petri dishes containing nutrient media where to reproduce. So use what we call the typical cultivation dependent method. So you had to reproduce, to help reproduce the cells in laboratory media that we call, this is just, for example, a petri dish containing culture media, and then take one of these colonies and then identify the species by a number of tests. For instance, what you see here is a number of biochemical tests that allows to isolate this colony and to understand that is E. coli, for example. Okay, so it was really a very, very long time-consuming process, and for an ecologist, it was really frustrating because we know that we have thousands of species in one single sample, so this approach was really not useful. And also, what was emerging in that year was that the environmental bacteria, so the bacteria that are here, if you go back in the lab and if you put here, this is just one example of a laboratory medium. For instance, it is a medium that is used to grow Vibrio. It is called TCBS. It is very recognizable for microbiologists, and Vibrio are these colonies. Only a tiny fraction of microbes from that water sample will grow here. So if you use this approach, that at that time was the only available approach, you are missing the big picture. So that was really frustrating. When I was a PhD student, like you, at the beginning of my thesis was on the relationships between biodiversity and functioning in microbial communities. That for me was really, really frustrating because you couldn't, it was like a black box. But I was also lucky because right in that times there was an emerging line of research, and I want to discuss with you because we have time, and I think it is really important to recognize the work that has been done. We have not to forget, because things evolve very fastly, and I think it's really important to know the history and of things. So what was emerging in that years was that that could be an alternative way to identify microbes, and we could use DNA. So there was no need to go through all these time-consuming tests. I can say that these tests, gram detection, for instance, or fermentation, whatever, it takes hours for one single colony, so it could be very, very long. And so there were pioneer scientists that were thinking, that were doing pioneer work to try to recognize and identify microbes from their DNA. And so the principle was that we could go back to our sample here to extract the DNA and then to identify bacteria directly by passing this time-consuming step. What I was not mentioning was that when you take the sample and we put a drop of sample here, it will take days to grow. So there is also one more step. And who was the father of this approach? Carl Boyce. Yeah, this is a very important scientist that was also awarded the Crawford Prize in 2003. If I'm not wrong, this Crawford Prize is something similar to the Nobel Prize for biology. If I'm not wrong, there is no Nobel Prize for biology, and they say that this is the similar prize, the analogous. But that was really a revolution because this guy was pioneering phylogeny. So he was suggesting that we could use a specific molecule of the nucleic acid, not only of microbes, but of all living forms, as a molecular clock. So we could use phylogeny to identify cells and to identify species. So it was really a revolution. I'm speaking about papers in the 1987. And then there were people who were trying to apply these approaches that were used in vitro. So they were using on culture, they were using on cultivated cells. They were trying to use on environmental samples. The target was the 16S ribosomal DNA. And this is the paper. So this is the first paper in microbial ecology where the first list of microbes using a cultivation independent method was used. If you look now at this paper, this is something that is really old, because now with the techniques that we have, the work that these guys did was really simple. But if we contextualize this, for that time was really pioneering. And so they were just doing this attempt using 16S RNA gene, but also 5S, so it's the smaller fragment, which is not used because the information contained is not so much. And then there were people that were benefiting of this approach. These are smart people that were publishing in Nature with the same approach. So if this paper is, I think it's 87 or 88, this guy, particularly Steve Giovannoni, they went into the ocean, they collected a sample in the Sargasso Sea, and they described the genetic diversity of Sargasso Sea bacterioplankton. This is something, Marco, that for that period, just one sample, Nature. Just think at now. Yeah. And for me, that I was reading years later, because that was, if I remember, 99. Yeah, I was still a high school student, but when I started in 2000, for me it was really wow, because there was a lot of technology, a lot of money, a lot of things that we couldn't reproduce in our lab. And also this paper is important not only because of the approach, but also because they were also opening the black box of marine biodiversity, of marine microbial biodiversity, and were also reporting something that we are still studying now. So you're welcome. Ok, perfetto, grazie. Adesso dovremmo esserci. Ok. What about the break? I don't know how you typically, maybe we can go for 15 minutes and then we break. Okay, so let's go back to Steve Giovannoni and his group. And so where they were really saying something that was also opening the way for many researchers later. So they were saying that in the ocean there is a lot to discover in terms of microbes, in terms of genetics. So if you look here, yeah, they reported novel microbial groups. None of the genes matched what was known. So was really something new. And which was the approach? So the approach was this one. It is something that now is considered old. We don't do this. I was doing in my PhD, so some years ago. And was really a lot of work. But now we have other approach that I will describe. But at that time, the approach was this one. So you had the seawater sample, which was here. You could take the water and filter on specific filters. Then you could take the filters, extract all the DNA from the filter that was trapping all the cells. I'm speaking about liters of seawater, so I'm speaking about billions of cells. Okay, remember the calculation. So just take 1 liter of water at the end, and everything is on a filter like this that has a pore size of 0.2 microns, so can trap everything that is larger than 0.2 microns. So basically what is passing is viruses and this organic matter. And then you could extract the DNA, Now it's very easy to extract DNA because now we have kits, but at that time it could take 2 days for extract just one sample, because it was a time-consuming process, because you had to extract the DNA and then you had to purify the DNA, so through a number of procedures. And then you could use the PCR. PCR also, that I'm sure you are familiar with, polymerase chain reaction. It is also something that has revolutionized biology. There is also very interesting story about the scientist who was discovering the PCR. I don't know if you are familiar with, but if you go on Google, just look at them. It was very crazy scientist. And it's funny because the paper of PCR was not accepted in Nature. It was sending to Nature. It was not considered important, but then it was publishing on, I think, methods in enzymology or something similar. But then it was probably one of the most important discoveries in biology of the last century. But then you could use PCR to amplify 16S rRNA gene, and so make copies, make plenty of copies of the 16S rRNA gene of bacteria. So you could make millions of copies. Then you had to run on the agarose gel just to make sure that the PCR was successful. At that time, you had to do this electrophoresis on gel, which was also quite time-consuming. Then you had to purify the PCR product, and then you had a single test tube like this, with 50 microliters, with millions and millions and millions of genes of all these bacteria. And then there was the need to separate them, because sequencing machine, they wanted just one single DNA. And so how to separate all these millions of molecules in 50 microliters of water in a test tube? You had to clone. You had to do what was called in vivo cloning and amplification. So you had to basically clone the fragments into plasmid. So the plasmid had to incorporate the fragments, that you had to insert the fragment into competent E. coli. So in laboratory you had to reproduce specific lines of E. coli and make it in simples, but it was weeks of work for one single sample. And then there was a kit that allowed to use nucleotides, which were labeled, every one with one single fluorochrom. So you had 4 different fluorochroms, and then you could run the electrophoresis. Marco, one sequence per time. One sequence per time, and then you had 90 clones, which was 90 different sequences of 90 bacteria from the original sample. And when I was like you, a PhD student, I did 4 clone libraries. I was the most happy PhD student of the world. But if you think now, it is something really. I'm just looking at Marco. Marco has millions and millions and millions of sequences from a set of samples. So there was really a revolution in that years. But that was at times. And the method was this one, was the Sanger sequencing, which at that time was even more modern, because the original sequencing was based on radioactive. So if you go in the 60s, the 70s, the first studies of genomes were based on sequencing that was using radioactive, so it was really toxic, complex, and also was producing a lot of waste. The Sanger sequencing was replacing the radioactive nucleotides with fluorochrom marked nucleotides, was this. And then at the end you had this wonderful sequence, and you could run into a software, and then you had all these peaks. that you can see here, and every peak was a nucleotide. Okay. Then there was a revolution. Which now is not a revolution anymore, but 10, 15 years ago was real revolution, which was next generation sequencing. Next generation sequencing basically allowed to sequence thousands, millions of genes simultaneously by a completely different technology. Actually, the technologies at that time were different. So there were pyrosequencing, there was Illumina, there was ion torrent. It will take maybe hours to explain them. I don't have time to go there. This is just to show you an example of the different technologies. But what I want to show you, and the concept that I want to transfer you, is that opposed to that method, you could sequence millions and millions of fragments. So it was a real evolution for all biology, of course, for the human biology, for biomedicine, and so on, but it was also a revolution for us, because remember that we had millions and millions of genes in our small sample. Yeah, here there are some slides that go into details of the technology. I will give you the slides, but I don't want to go through too much on this. I just want to show you how these revolutions can be quantified. I remember I was at the beginning of my PhD that there was this announcement of the first human genome. So there was the sequencing for the history of the planet of the first human genome that was completely sequenced. That was the result of a very big project funded by the Americans, of course, the richest. And it costed $3 billion, one genome. 13 years of work of hundreds of people, tens of different research groups across the US. So it was really an immense work, which was because it was based on the Sanger technology. So it was 300,000 genes, but they had to be sequenced again. This is 2014, so I think the cost today is less. But if you wanna sequence your genome, you could do in a few days for hundreds of euros. So from billions dollars to hundreds of euros. So the technology has completely changed things. And based on this, there has been a new discipline, which is called metagenomics, that has revolutionized the study of the environmental microbiomes. So metagenomics, there are many different definitions. Is derived from the Greek, so meta is beyond, and to make it simple, it's something that studies beyond the single genome study. So the concept is that in the past, before next generation sequencing, you could study single genomes, single genes, single organisms. Now you can study meta. Maybe if we think of today, metaverse, so it's the same probably. So you could study the whole of the things. And now, if we think to metagenomics, in particular in microbial ecology, so it is typically referred to the study of all genes from environmental samples without cultivation. So you can take a sample, seawater, sediment, epibionts, air as well. We have microbes in the air that we are breathing, surface, biofilm, or whatever. You extract all the DNA, you sequence, that is metagenomics. And that was also a changing paper by Rob Knight, because Rob Knight was the first with this paper that was also showing a bottleneck. So the technology was impacting a lot. It was apparently easier. It was a new golden age for us. But then there was also a first bottleneck, which was the amount of data that we generate that takes time and machines to analyze. Because the amount of sequence that we were producing now was so big. So we were entering in the big data of marine microbiology. And this was also a bottleneck, which is a bottleneck of today, because every time we propose new fundings, all my colleagues also think, okay, we want to study this, okay, we need this amount of money for sequencing. And then I go and say, okay, but also you have to add man-months of people processing data, because it's not easy. I don't know if I can, there's no, okay, there is no phylogenetic tree here, but phylogenetic trees for the first papers with Sanger sequencing were very, very easy to see. Rob Knight was showing things like this. So a typical sample which is analyzed show a phylogenetic tree which is impossible to interpret, because this is a typical information that you get. So it's working. Yeah, so this is just one sample. Yeah, it is. Yeah, just at the beginning here. Okay. And it's funny if you just think that it's just different combination of 4 letters. It's a lot of work. So I think we can stop here and have a break, and then we will go. If you have questions, of course, ask or later. Okay, can start. So that was the list. Okay. And that's the challenge. Okay, so the challenge today is to how to analyze the data, to classify them, to make tables of taxa. We use the term OTU, which is operational taxonomic units, that now is also, there's another one that is called ASB, sequence variant. So it is a really dynamic and evolving field of research. But that was just to show you an example of the picture. And this also underlines the need to foster multidisciplinary science. So bioinformaticians and ecologists together. Because bioinformatics is becoming complex. Maybe some of you will work as bioinformatician in their career, I don't know. In my time, because of the number of sequences was so low, you could be an ecologist and bioinformatician together. I did all this work for my PhD thesis, but now things are becoming complex and complex, softwares are complex, big data are complex, and so this is why it is important to favor the synergy between ecologists and bioinformaticians. Okay, that was a more technological sections. I hope it was useful for you. Sorry if I was pushing a lot, but I wanted to explain all the history. Now I want to move forward, and I will speak something about the microbiomes in aquaculture, which is also part of the work that I'm carrying on my research. And to do this, I just wanted to make a framework of where aquaculture is in the international context. Sorry if it is in Italian, but if you look at the context of marine ecosystems in general at the planetary level, we know as scientists that situation is not very good. So if we would like to have healthy marine ecosystems, because we are learning and we are trying to learn, politicians and stakeholders, that we really need healthy ecosystems, but not only because we want to swim in clean sea, but because the health of the ecosystems, the health of the seas and the oceans is also our health, because healthy seas really support life on the planet. But what we are looking is that the human imprint on marine ecosystems has grown in the last years. This is a very nice paper published in Nature a few years ago by Carlos Duarte, which is particularly dedicated to marine life, so how marine life has been overcatched, overused, overpredated, and which were the steps at the political level to start protection, and also they try to understand and to explain how rebuilding marine life in the oceans should be done, and could be also profitable in terms of more fish to be fished, in terms of conservation efforts. And this is also important to understand which are the planetary boundaries for the blue planet, because we know much better the planetary boundaries for terrestrial ecosystems. This paper also, and I suggest to read, also integrate the planetary boundary concept also for the marine ecosystems, and indicates which are the boundaries that we have not to surpass, because otherwise humanity will be in crisis. And if we think at fish and aquaculture, we have to remember that the food from the sea will be even more important. Actually, the fish is not providing a significant source of protein for the global population, but the demand of fish, the demand of food from the sea is increasing. And of course, we are getting fish we are getting food from the sea by means of fishery and by means of aquaculture. Fisheries, in this room there are more experts than me, and but we know that fishery is really important in terms of the economic importance for the society, in terms of the workers that are employed, in terms of the food that we have. But we also know that fisheries is in crisis, because many stocks are not in good health, many stocks are overfished, and of course we have to work a lot on stock assessments. We have to produce, we have to move toward a fishery that is more sustainable for the environment and more sustainable for the stocks. And if you look at the aquaculture, which is also growing a lot, I will show you a graph where it is shown that aquaculture is defined as one of the most growing industry at the planetary level, because in the last decades aquaculture has grown a lot, and this is also because the demand of food from the sea is increasing, also because we have to support an increasing population. Aquaculture can be an important alternative to fishery, but also aquaculture has to transform itself, because aquaculture has to operate much better than done so far. Aquaculture has to operate under the One Health perspective. The One Health perspective means that it has to be preserved the health of the farmed animals, the health of humans, of course, but also the health of the environment. So under these three groups of three sectors, aquaculture has to transform, and I will show you some numbers. So we are, humans are growing and growing and growing and growing. We will be 10 billion people at the end of the century. And if you look at the official numbers, these are from the FAO State of the World Fisheries and Aquaculture. The food from the sea now is providing just 17% of the meat, let's say, okay, of the global meat. But it can be higher in some countries that are more dependent on food from the sea. But it will increase. It will increase a lot because there will be increasing demand of food from the sea. The total production today is 214 million tons coming from these two industries, so from fisheries and aquaculture. And from these, 9 million tons are coming from fisheries. So aquaculture is providing more food than fishery. As I was mentioning, the consumption of aquatic food is today 20.5 kilos per capita. This is an average, of course. This is projected to increase, and it was less than half 20 or 30 years ago. So it is increasing. And also many fish stocks are fully or overexploited. So this is a global important problem. We know, especially in the Mediterranean Sea, there are many groups working on this. And also, as I was mentioning, rebuilding overfished stocks could be an option, of course, and there are estimates that it could increase the production by about 60.5 million tons. So if you look at the global trends, you will see that this is the fishery here captured from the 70s. So you will see that there was a continuous growth, and then the production is stable. While if you look here, this is the aquaculture. Aquaculture in the last years has grown a lot. We need food from the sea because they are really a good food, because they have a role in food security, they have a role in nutrition, they are a source of proteins, as we say, but they are also a source of a lot of other molecules that are also useful for us. So omega-3 fatty acids, micronutrients, and so on. And if you look at the growth of aquaculture, there has been an important growth in particular regions of the world, particularly Chile, China, and Norway, because they are top producers in particular types of aquaculture. Chile and Norway, of course, for salmon, but also China is growing a lot, and Asia in general. And the experts are saying that predictions is that aquaculture will double its production in the next 20 or 30 years. So aquaculture will become more and more important, and also the European Union is pushing a lot aquaculture also as an alternative and also as a way to reduce the pressure on our fish stocks, that are in many cases overfished. But as I was saying, the aquaculture, this is the vision of the EU, has to transform itself working on a number of topics where there are plenty of works for research, for enterprises, and so on. Aquaculture has to reduce the footprint, so has to move toward a lower carbon economy. And this is also part of a lot of things. Let's think about the feeds that are used. We have to reduce the footprint of feeds in aquaculture. Aquaculture is sensitive to climate change, as fishery is sensitive to climate change, because we know that fish are extremely sensitive to the changing conditions of our oceans and seas. So fish populations are moving. Okay. Seawater is warming, for instance, and this is also affecting the physiology of species. Aquaculture has to reduce pollutions. We know since decades. I was publishing on the pollution of aquaculture more than 20 years ago, but we know that intensive aquaculture has an impact. It's not devoid of impact on the surrounding environment. During my PhD, I was working on an EU-funded project that was, for instance, studying the negative effects of aquaculture on marine plants, on Posidonia oceanica, for instance, because accumulation of fecal pellets, organic matter, and feeds on the seafloor, where Posidonia oceanica was living, had very negative effects because it was causing enrichment of organic matter, anoxia, hypoxia, and so on. That was just an example, but we know that aquaculture is producing a lot of negative effects in terms of eutrophication of the surrounding area, use of chemicals. At that time, the use of antibiotics was massive. Now we know that the use of antibiotics is changing. If we go to the supermarket, we buy sea bream that is without antibiotics, for instance. So many enterprises are moving toward antibiotic-free productions. We still have problems. We have problems of accumulation of past usage of antibiotics in the surrounding. So we have many impacts of aquaculture of the surrounding environment. So aquaculture has to transform and become a zero pollution aquaculture, as the strategy of the EU to zero pollution is taken to 250. Aquaculture can be also biodiversity friendly, so can have less impact on biodiversity, so reducing pollution, reducing the use of animal sources in the feeds. So we can use alternative feeds. For instance, many feeds are derived from fish, but we can use alternative ingredients. We can use microbiota. For instance, I will show the results of our project as probiotics. We can use insects. We have been running some projects in Messina for this, but there are many examples. And also aquaculture can become circular. This is also true for the fisheries, but for aquaculture as well. We are discarding a lot. We have a lot of discards in terms, especially during food processing. So we have to think at new ways of reusing what we waste from aquaculture. There are many examples, and what we are not eating, what we are not using, can be valorized, and by creating also new company. And this has to be reflected in the One Health aquaculture. This is where all the efforts has to move. This is something, just a summary of what I was telling to you. Here you can see some references. for the use of antibiotics. Vaccination, for instance, is also increasing a lot, especially on salmon. They are doing a lot of work on the use of vaccines. There's a lot of research on how to treat disease, because disease in the aquaculture can be a problem because of the farming conditions. They live in very close contact, so if you have one diseased fish can infect all the surroundings, so the spread of disease can be very fast. And also the use of antibiotics. One of PhD thesis is about the use of antibiotics. Now, if I'm not wrong, so it is, for example, a EU-funded project which is called ARENA, and it's just studying the fate and the destiny of antibiotics-resistant genes in the aquaculture environment. So we can be also legacy from the past, but also like in the hospitals, you know. Yeah, have you been hearing about the spread of multi-resistant infections in our hospitals, for instance? As our hospitals, aquaculture sites can be sites where antibiotic resistance can spread. It can also spread to the natural community. So we have to learn. We have to develop methods for detection. We have to develop sensors. We have to develop rapid method also to detect presence of antibiotic, but also antibiotic resistant genes in the fillet, in the fish. So this is also a line of research. And this is the big picture I was showing. So how pollutions affect the surrounding environment. Also the spread of escaping fishes can be a problem of genetic pollution, because they can mix with the natural population, so they can create problems to the wild populations. And the use of feed, the use of chemicals, the spread of parasites. So as a director, I have to say that at all the tables I have the privilege to sit, I see a lot of attention by our governments, by the policies, by the funders for aquaculture. And this is also can be an opportunity for you in your future job. But I see aquaculture in the future as a real stimulating sector that will get a lot of funds, because there is a lot of work to do for a better aquaculture. And then let's go back to the microbes. Because if that was the big picture for aquaculture, one part of the big picture is the microbiome, because microbiome are part of the picture. I'm speaking about the fish microbiome. I'm speaking also about the marine microbiome, because when we deal about pollutions of aquaculture on the seafloor, pollution can be mitigated by the resident microbiota that can degrade and transform the fecal pellet or the feed which is not eaten by the fish. So all the microbiome in aquaculture is also something that has to be studied and also can be engineered for a better and for a more productive, for a safer aquaculture. So this is the definition of the microbiome that I was giving to you at the beginning of the talk. And so just remember the difference between the microbiome and the microbiota. So they are two different terms. Don't use them as synonyms. It was born for studies on humans. This is also our frustration as an environmental microbiologist, but as was for next generation sequencing, for cloning, for everything. Things first are on the biomedical side, and then we take and we transfer to the environment. So actually the concept was born on humans, and now is used for all the environments. There is also a journal that is called Microbiome. As I was mentioning, they have a role. They have a role that we are starting to learn. Okay, most of the work has been done on the gut, because the gut is the place where many microbes are living, because the gut for humans, but also for gut-having animals is important, but there are also many other tissues that are important to study. And this line of research is growing. And this is a book that was published a couple of years ago, which is showing some updates on microbiota research. So microbiome research in the last years has been growing, because basically the role of the fish microbiome can be really important for the fish, but it is really diverse, because the fish microbiome can give protections against pathogens. It is also for us. If our intestine is, if the microbiome of our intestine is in good condition, it is more resistant to infection. It can help the host immune response. It can also be important for the nutrient acquisition in the gut, of course, but also in the gills. The gills of the fish that have a role, that it can be also important. It has a role for the growth of the fish, for the development, but also can help in tolerating antibiotic stress. So all these roles by the microbiome are important. But at the same time, if you see here, these are the drivers of the microbiome. So microbiome will depend on the host genotype, as we are learning, but also on the host environment. And this is particularly true for marine organisms that live in seawater, so they are in very close contact. So just imagine how many interactions there are between the microbes that are on the surface or on the gills of a fish and the surrounding environment. Okay, so this is real very dynamic interaction. And we are just trying to dissect this interaction. So these are just examples for you. I suggest reading a lot during the PhD. For me, it was important. Read everywhere, every time. And there are plenty of papers that are opening this black box of the fish microbiome. And the next step will be, we are still in a phase that we are describing, but the next step will be how to use this knowledge to make aquaculture a better industry, to make aquaculture safer, to make aquaculture more productive, to make aquaculture more resilient to climate change. These are some of the hypotheses, but was basically already said. And the numbers are these. Of course, they are just range, because there are variations. So the skin is a place where there are 10 to the second, 10 to the 4 bacteria per square centimeter. The gills also have a lot of bacteria. And as I was saying, gills are still a neglected part of microbiome fish studies, but we think they are really important, because they are at the interface of the environment. And the gut, where many studies have been, of course, dedicated to, where we can have up to 10 to the 8 aerobic heterotrophic bacteria and up to 10 to the 5th anaerobic bacteria. So the gut is really the hotspot, the hotspot of microbes. Skin, gills, olfactory systems, they are the first contact points, so they are important. The mucus is typically also a hotspot of microbes, because it can trap microbes, but also can provide food for the microbes. So it's rich of organic carbon, dissolved organic carbon, and so there can be many microbes that can stay there and eat, because it's a place where there is a lot of food. Then they can be also barrier for pathogens. This is also something which is important to understand, because this is like ecological theories. There can be an ecological niche. If the niche is occupied by other microbes, it can be there could be no space for other microbes. But of course, this is also the results of interaction at the micro scale. So it is really difficult for now to really understand what is going on when you have gills with plenty of microbes and other gills are coming. So there will be a number of interactions of different chemical sensing. It will be something that's still to be deciphered. But this is the theory. The theory is that a good microbiome of a healthy fish can be a primary barrier for other infectious agents. These are studies that we found in literature. For instance, for the gills, we know that the community of the gills differ from the skin, and this is also confirmed by our studies. I remember there are some authors that also were thinking at studying the gills microbiome as a tracer to study microbes, just to understand where the fish have been swimming, because they could conserve some microbes that the fish can have taken during fishing. That could be also an interesting hypothesis. But also what we are seeing from our studies is that the gill microbiome is quite stable. It's quite different from the gut, it's different from the skin, and it is made up by a very different community. This is the gut. The gut is a complex tissue, so there are different compartments that you see here. I don't want to go through in detail of this, but the first studies have been done on salmon, of course, on mullet, on eel, and there are different studies. A study that has been published that we are also replicating for sea bream is that the microbiome changes during the fish development. So if you start studying eggs, the different larvae, the adults, and so on. So if you go through the fish development, you will see that the fish microbiome changes. And this is important, starting from the fish egg, because the microbiome of the fish egg is obtained by the mother, by vertical transmission. And this is something that is also known for humans, for instance. And then from the egg to the larvae, and so on. There's an echo. The microbiome is changing, and understanding how this microbiome influences the successful development is something that is being studied, because that could be changes in the microbiome that could lead to changes during the early development of the fish. Yeah, the first studies along the fish development are showing that microbial diversity from larvae to adult decrease. We know that diet is important in colonizing the gut, especially during the growth. There are a number of taxa, for instance, bacteria and so on, that we are now learning that they are common, typical of the gut, but how they function is really don't know. And what we suggest, especially in this paper, this one that we were publishing last year, is that we have to move beyond the description of the microbiome and integrate biotechnological approach to understand how these microbes play a role and to understand which role they play. So to do this, we have to develop new approaches. For instance, biotechnological approaches such as gut on a chip, synthetic guts, synthetic communities in vitro, or the use of germ-free fishes, for instance. All this information has to be done in order to understand the role of this microbiome. There is also an effect of the environmental conditions. We have published a paper that I think I can show later. And because seawater condition changes in different conditions, because they reflect also the physicochemical condition of the environment, there is also an hypothesis that the microbiome associated to fish can change. And this is also something that we have to better understand, not only for the fish, for the aquaculture fish, but also for the wild fishes, because as the oceans are changing, the physical chemical conditions are changing. We don't know how microbes will be affected by these changes and how the interactions with the holobionts will change. The diet, of course, has a role, and we are carrying out an experiment that I'm showing. So diet changes a lot, and this is why it is important if we want to develop new feeds for aquaculture to study also the effect on the gut microbiota. And this is just examples that are taken from this paper by Egerton. It is on Frontiers, and you can just go on and see how are the species. Just to mention that many of the species are based on cultivation, so maybe now you know that cultivation-based approach and DNA-based approach can be different. Many groups are still working on cultivation because cultivation is still important, because what I was forgetting to say is that the power of cultivating microbes is that we can have the cell in a tubes, and so we can have the organism and then we can grow and do the test. With DNA, you just have the DNA sequence, but you don't have the bacteria to do manipulative experiments. So I don't want to say that we have to abandon cultivation. Cultivation in microbiology is still important, but if you want to do a list of species, if you want to describe all the species without any bias, metagenomics is the best. These are still a general view of the role that the microbiome plays for the immune systems, for the physiology, because they can do plenty of roles. They can facilitate digestion of food, they can help degrading molecules, they can help degrading complex molecules. And this is also because, as I was mentioning, and that's the same for us, and that's the same for environmental biotechnologies as well. Microbes work together in consortia. So this is why if we get probiotics, if we take one species, it's nothing, because we know from the environments, we know from the gut that consortia are really the key, because there are many species, and because the synergy, because the products that are degraded by one are then degraded by the other, and so on. So communities are important, and this is also the case for the degradation of recalcitrant molecules, which can be degraded by complex consortia. That's the same for bioremediation, for instance. Maybe you have studied at university the first bioremediation to degrade hydrocarbons at sea. There was the first strategy was to go there and put the strain of alkanivorax or hydrocarbon degrading bacteria. They don't work in single species, they work in consortia. And of course, the feedback is that the diet changes the microbiota. So a more diversified microbiota can help degrading better feeds, but also the type of diet influences. And wild fish microbiome, I was at Aqua Farm, the largest aquaculture expo in Italy a couple of weeks ago, and I got the question about wild fish microbiome because we are studying in Marco's thesis the microbiome of wild sea bream, which is quite appreciated because we have very few data on wild fish microbiota for a number of reasons, also because it's not easy to go and get wild fish and study the microbiome, because if I go at the shop, the microbiota maybe has changed because it's not a real freshly collected. And if you get from fishery, you can go on board of fisherman boats, but for some species that are not the target of trawling, for instance, it's not easy. So there are a number of reasons why the microbiome of many wild fish is not known. And so it is important to know and to study this. And what is emerging also from our studies is that the microbiota is different, and it makes sense because they eat different things. Yeah, this is a review, which is quite a landmark that show the studies of microbiome. And it's growing a lot. So the first studies, this is also paralleling the growth of NGS sequencing. So at the beginning there were very few studies. In 2018 there were 40, now I think there are hundreds. And as I was saying, microbiome could be important for the aquaculture, can be important for the aquaculture. And this is, there are many tools that can be used. These are more or less the tools I was mentioning, so NGS and cultivation and blah, blah, blah. And now I'm moving to the last part of the talk, which is where I want to tell you more something about the project I am following, that is called Circles. That is a typical example on how to use microbiome for a better and a safer aquaculture. So the project is a EU-funded project, is funded by Horizon 2020. One of the last calls actually was starting in 2018. You know that Horizon 2020 has been a very important funding project by the EU, and it has now been replaced by Horizon Europe, that has started recently and will last for the next years. I'm talking about tens of billions of funding, so it is a very important source of funding for all the EU. And I remember the Circle project was when it was funded, there was a specific call which was dedicated to improving knowledge of microbiome for sustainable food production. So it was not only meant for aquaculture, but the EU wanted to promote research on the microbiome to move for a more sustainable food production. And we applied to this project with Marco Candela, a professor, as a coordinator, and we'll build up this consortium that you see here. So it's a typical, you are probably familiar, or you will become familiar on how to build up a research project. So basically there is a call with a topic, and then you start making calls and emails and looking at your contacts and thinking how you can build a competitive proposal. Particularly Horizon Europe and Horizon 2020 are very competitive, so typically there are many applications for just a few lucky projects that are funded. And so you typically have months or 1 year or 1.5 years, depending on how the fund is relevant and how complex is the call, to build up the project, structure into work packages, into objectives, and so on, and then submit, and then the proposal is passing through international reviewers and then is funded or not. So this is what we did in 2017, and the project was funded in 2018. So it's coordinated by Unibo, and then there are other academic and research centers, and then we had many private companies. So this particular call that was dedicated to sustainable food production wanted to have private companies or small medium enterprise. They use the term SME. We have SME, but also very big companies. We have, for instance, what is it? A Gruppo Veronese. Here we go. And then we had Euromix, for instance, which is an enterprise that is working on environmental bioremediation, and many others. And also we had, I don't remember which one, we also had a company involved in dissemination, which is also now an always increasing part of every project. So dissemination, explaining to the society, to the stakeholder, all the achievements of the project. So circles want to study the impact of food systems, microbiome, on the main European food system. So basically subject of circles are poultry, chicken, salmon, and sea bream. These four animal food chain are the real pillar of the food systems of Europe, and this can be done, I would say, for the first time with a dedicated approach through the microbiome. Of course, I will speak about sea bream, which is the core of our research at CNR. And the paradigm is that in circles we want to observe, we want to compare, and then we want to intervene. So basically we want to learn more, and this is just to fill the gap. As I was mentioning, we know something about the fish microbiome, but it's not sufficient. We have to learn more. And then we want to compare, and then we want to intervene. So the last part of the project that has started right now is to manipulate the microbiome in our tanks and see how aquaculture is changing. So in terms of observation, what we have been done very quickly is to study the microbiome dynamics all across the life cycle. As I was mentioning, there are some papers already, but we are doing again because for sea bream we have to learn also the role of the environment where the larvae, the eggs, and the adults are living, and so we want to produce more knowledge. And this is currently done. We have the data, but we are still analyzing the sequencing, and this was done in a fish farm in Sicily, collecting basically starting from the eggs to the larval stages to the adults. Then, and this has been already published, then also we were studying adult farmer sea bream, their microbiomes and the relationships with the surrounding environment. This was also already done. So what we did was to study the microbiome of adult sea bream in different tissues, so skin, gut, gills, and also fillet. Of course, we know that the fillets should be sterile, but also we found some some presence of microbes, and so we are still interrogating if they have a role there, they are a simple contamination. So that was an open question. But also what we did was to study the microbiome in the surrounding environment. This is something that is not done very often, so study in the water and in the sediment. And what we found was that, of course, the sea bream microbiome is distinct. The microbiome differs between the different tissues. This is something that is related in this picture, which is a network analysis. And you can see that the different colors relates to the different tissues or to the different environments when we talk about seawater and sediment. Also, one finding was that the sea bream microbiome reflected only partially the one from the surrounding environment. So our conclusion on this data set was that the host is the primary driver. So if we go back to the hypothesis, if the environment or the host is the primary determinant, our data suggests that the host is the primary driver. There were some taxa that were coming from seawater, from the surrounding environment, and we also find that the influence was much with seawater than with sediment. And this similarity was especially with the gills. And it was also quite simple to explain, because as we know, the gills are really the first contact point with the surrounding seawater. As I was saying, we also made a lot of effort in collecting wild sea bream. It's not easy. You see our efforts here to collect wild sea bream. This is also an open question in the community, because one of the questions I had at the conference a couple of weeks ago was why we decided to get wild fishes very far from the farm in Sicily, because we were collecting wild fishes in the Adriatic. And of course there was a huge discussion with the partners, because I was forgetting to say that the work we are doing in Sicily is also done in Greece and is also done in Spain by different colleagues. So the strength of Circle is also that the same studies performed at the Mediterranean scale, and then we will take the results together just to see if the results are reproducible or not. And one of the discussion was how we can consider a wild sea bream truly a wild sea bream, because we know, and we had one big example in Ligurian Sea right these days, if you have problems on a cage, there will be a lot of fishes escaping from the fish farm, and that will contaminate all the sea bream population in the surroundings. So even the experts in fish genetics are really, they don't have good tools to truly understand or to truly state that the sea bream is wild or is not coming from aquaculture, because of the mixing of the population. So our choice was to go quite far. So we collected in different seasons, in different locations of the Adriatic Sea, but far from the fish farm. And we also were lucky because we had a colleague who is very skilled in collecting sea bream. It's not a joke, but it's not easy to get wild sea bream and to process them immediately without changes post-mortem to the sea bream. We cannot go to the shop. We cannot wait for a sea bream to come hours after collection if we want to really study the wild microbiome. And so it is not easy to know when and where to get adult sea bream. So this is also explaining why the literature is missing real data, and this was quite appreciated by the community. Then the comparison. So this part it's quite complex. I will try to explain, but it was basically a bioinformaticians and modeling exercise. It was done by colleagues in Unibo. So you remember the paradigm is that we want to observe, we want to compare, we want to model, and we want to intervene. Okay, so ideally the project wants to describe the microbiome of our sea bream, and then we want to try to identify which is the perfect, the more healthy microbiome of farmed fish. It's not easy because we don't have a reference. We don't know how we can define a fish microbiome as good or bad. So what we try to do, of course, it's a way, it's not perfect, was to hypothesize that the wild microbiome, wild sea bream microbiome can be better than a farmed sea bream microbiome, because the farmed sea bream microbiome is manipulated by the fish, is eating different things, there's a human imprint. And so, basing on this assumption, we were comparing the wild sea bream microbiome with the farmed sea bream microbiome. It's a lot of work that I'm synthesizing here. I even just remember the table with millions of sequences, but at the end, using appropriate tools, what we could find was to identify a number of taxa which were enriched in the wild sea bream or enriched in the farmed sea bream. So there were differences, particularly with some taxa that were then clustered here in this. Are clustered here using network analysis, and at the end, to make it simple, that was really not easy for me to explain. It was a work lasting months. Is that we could identify some genera that could be positive or negative for the fish. It's not the perfect approach, of course. It's not easy to do microbiome manipulation, because also using probiotics is not easy in aquaculture, because there is a very strict regulations, because there are it's not complex. Sorry, it's not easy. So what we decided at the end was to identify which taxa could be positive, could be beneficial for the fish, and basing on the literature to identify molecules or complex that could stimulate the growth of this taxa. If you want, I can repeat, or I hope it is clear. If it's not, please forgive me. But at the end of this reasoning, what we did was to identify some components that we are now testing in our tanks in Messina. So they have been added to the feed, and they are caprinic acids, short-chain fatty acids, tannins that are extracted from trees. Basically, they are extracted, they are vegetables that are extracted from some trees, and some prebiotics. So, as a modulators of the feeds that we are currently using in our experiments. This has been done with the industrial team that I was mentioning. So Veronesi is a big producer and partner of our project for feeds. Silva team is a very good company, Italian, working on tannins. I didn't know anything about tannins. I was learning in circles. Tannins are complexes of compounds that are extracted from trees and that are used in industry for many purposes. And they can be also used as integrators because they have stimulating effects or inhibition effects on microbes. So they are really promising. And I think there is a PhD study on this. One of their colleagues, maybe if I'm not wrong. So this is our plant, our facility at CNR in Messina, and this is the description of the system. experimenting with fish in aquaculture is not easy. There is a very, very complex regulation. It's not like working with corals or with invertebrates. So the EU regulations on animal experiments on fish is quite strict. So you have that your plant has to be approved by the Ministry of Health. And then for every experiment you have to pass through a quite complex clearing procedure. So you have to plan your experiment. It was planned maybe 1 year ago, and you have to clear through a number of regulations. And when you have permission, you can start. And this is the feeding track. So we have a number of authorization by the ministry, which is done specifically for our experiment. And we have 476 gifted sea breams in our tanks. This is the initial weight. This is when the experiment started last November. And these are the diet that have been produced specifically for the project by Veronesi. So we have one commercial diet. That's the one that is typically used by the fish farm, the fish producers that you buy at the supermarket. Then we have a control diet, which is enriched with fatty acids and tannins. Then we have another control diet enriched with prebiotics and tannins. We give daily, and then the experiment is going on. Then we have some trial of 30 days, 90 days. The experiment will end in June 2023. We have, of course, a number of replicates and so on. And what we are also measuring is not only the microbiome, but also a number of variables that we want to use as indicator of welfare of fishes, of productivity, of quality, of physiology of the fish. And this is particularly what we are doing, also in collaboration with CIMAR, which is a Portuguese research center. So we study microbiome in all the tissues. We will study Mazzara del Vallo, the quality of fillets, the nutritional composition. So how this different diet that has an effect of microbiome will also affect the fillets in terms of nutritional quality. And also during the experiment, the growth performance, the health of the gut by histological analysis, indicators of stress, immunity, and so on. This is just an example of the teamwork, just to show you how a complex experiment like this that I was simplifying in 2 slides involves very multidisciplinary team of workers and also a lot of efforts. And yeah, these are some useful reference for you if you want. Just the last slides, just to say that I've been dealing a lot with microbiomes and its role for the teleost, but of course all what I have been showing, including the methods that I was explaining to you, so next generation sequencing, DNA sequencing, and so on, can also be useful for another research line, which is not the subject of my research, so I will go very, very quickly, but I just wanted to mention you, is that the food safety. So the food safety in terms of disease that can be associated with fish, with eating fishes. And of course, we know that in general, climate change is affecting infections of marine animals. This is well established now because the changing condition of our oceans and seas, and particularly warming of temperature, increasing of nutrient spread of animals, spread of alien species, pollution, increasing sewage that we that we deliver to the oceans, all these pictures together is leading to an increase in infection rates of animals in general, of marine animals. So we have many famous cases. Just think about coral bleaching, whitening some years ago, but we have many infectious diseases that are affected. And this, of course, has also implications for the safety of food and the safety of fish. And this is, of course, this is another topic. So there are many groups studying the spoilage of fish product, and I guess that everybody in this room is eating sushi as well, and this is also something that is important because it has been a deep transformation of our habits. I eat sushi as well, but of course it needs also a lot of research to do in terms of how the fish are treated. This is from the microbiology point of view. This is not only the point, but from the microbiology point of view, we have to monitor, and we also have new tools that we can use. So there are regulations from the EU. It's a very active field of research, but I don't go through too much because it's not my expertise. Okay, I should have finished, but we can have an open discussion if you are not too tired. Don't be shy. Okay. If you don't have, it's okay.
I have a question. You told at the beginning that there are multi-drug resistant bacteria and so on, and I know that also in aquaculture a lot of drugs are used to treat fish and so on. Have you in mind to study bacteriocin and producing strain that you use in aquaculture to avoid the use of antibiotics or not?
We are not studying as a research group, but yeah, this is also a line of research that is growing, and also I also remember that one exciting line of research was the use of phages, bacteriophages, to fight infectious disease. And I remember some very interesting paper, and I was not director, so I could read much more than now. And but that was very promising, yeah. But I have to say that now antibiotics are still the most used drugs because they are cheap and they are effective. Yeah. I mean, many of the sea bream that we buy now in the big supermarket are from very big companies that they are antibiotic-free companies, so they are certified, and I can say that with respect to the past, the use has decreased. But not all of them, so I think that also the consumers are not very aware of it. They just go to the supermarket and buy. They don't even know when they buy a sea bream if it is farmed or not. You know, so there is a lot to do also in informing society. Yeah, but also the project that I was mentioning, which is part of Naomi PhD, which is the core of Naomi PhD. Also, one output that we have will be, which is called JPI Arena. So it's funded by JPI, so it's a joint programming initiative in the EU. And one of the output that we really look forward will be also to detect methods and tools to measure rapidly residuals of antibiotics in the fish fillet, and also to measure the presence of antibiotic resistant genes. So that would be that's something that the company wants to. That's it.
About the use of this kind of study like microbiota, how to know the general condition of an area in which the fish live, for example. It's possible to use the microbiota to infer about a polluted area, or, for example, thinking about
Fishing effort.
But also, pollutants.
Yeah.
Pollutants, for example, comparing the microbiota of fish living in a marine protected area, for example, what do you expect to find? It is a good question. I don't know, but I am curious about even from this point of view, it is possible to recognize particular group of bacteria living in wealthy fish compared to the stressed one.
Yeah.
Both from a point of view of fishing pressure, but also from environmental pollutant, for example.
No, that's a good question. And
is there are there some paper about this topic?
I think the papers are emerging because this is the next step. You know, how to use, for instance, microbiome as an indicator. And I remember that someone was suggesting the gills microbiome, because the gills, as they are at the interface, could have been trapping bacteria that are associated to pollutant areas, for instance. We were discussing just a few days ago if we could link the microbiome of wild fishes to fishing effort, for instance. So there are plenty of things that can be done. Marco's first data on Solea, for instance, they're showing that the skin that has a lot of mucus and it's swimming in the sediment has a lot of sedimentary bacteria on the mucus, and the gut is completely different, of course. And so that could be, for instance, a good candidate, because I understood that the Solea is swimming a lot. I didn't know. I thought that it was staying there. But I understand from the fish expert that Solea is swimming a lot. So that could be a good candidate, for instance. I mean, there are many opportunities that has to be done. Marine protected area, for instance, that could be interesting to. We have to still achieve the optimal microbiome for target species, and then understand if this is done for humans. There are plenty of companies now that if you have problems, acidity or whatever, take a sample and they say that you have dysbiosis. Okay, so your community is not in good order because maybe you have more firmicutes, or you have relevance of certain taxa that compared to a healthy microbiota can be different. So for humans, this of course is already been done. For fish, we are starting to do. That could be. There are plenty of opportunities to link to pollution, to conservation. Fish's effort is something that we are working on. That could be a very, very interesting things to demonstrate that fishing effort has an effect on the microbiome. Maybe because of the stress, because of the alteration of the physicochemical condition of the sedimentary environment, because we know that trawling is changing a lot. No? We don't know. Let's see.
Yeah, or even the increased number of anoxic events.
Exactly, yeah.
In the last 10 years, for solar, you know, the other part, almost every year, fishermen come to us and say, we are catching a lot of solar already dead.
Yeah.
And then, for example, to Tesaro, we send some samples to some colleagues of the University of Padua to understand from with your approach, no? Because the only way is to understand that bacteria were already present before the diagnostic period or after.
Yeah, also one option could be to understand. I know that people working on fish stocks, they have problems in defining fish stocks using genetics. So maybe microbiome could be an option to explore. Maybe we can distinguish Adriatic solea stock from, I don't know, a GMC basing on the microbiome. In that case, I would suggest the gut because maybe it's more stable. But yeah, that have to be explored.
So, following on this topic, that is you say that you have some preliminary results on those soils. I am very curious to know about the stability of the consortium of the bacteria, for instance, in the gut. For something soils in a area as small as the other cities, we saw differences in the gut content between the intestine just by eye.
Yeah.
So I'm curious to know if you recognize stability in some not respecting the areas, but something that wasn't out of the consortium some part, because I don't expect stability on part.
Yeah. Yeah, that's a good example of synergy between fishery science, microbiome science, yeah. And yeah, Marco is writing.
I'm still analyzing the data because it's not easy to understand which variable can affect more maybe the gut or maybe the skin. Just we see that the preliminary data that is different from skin to gut, but we don't know from the gut is that if the difference is inside between the gut is derived from area or stage, development stage, maturity, but we will see maybe with more data.
Exactly. Yeah. Yeah. I know that in IRBIM, that's not a limiting factor to get the fish.
It will be the basis to associate the microbiome with
Yeah, as I was mentioning, the bottleneck is data analysis, because it's you need effort and also you need computing power in terms of and time to interpret, because also the data that are produced are so big. And yeah. And then I only showed you 16S studies. So basically we are focusing on just one gene, that is the 16S that describes the species. But if you move to metagenomics, so if you move to all the different genes, not only for taxonomy, but also for the functions, so you can sequence everything and then you have to classify and see, and that's really huge work we are doing in Circle, and it's really promising because you get all the information, but then you have to interpret and to cluster. Stefano, I was finishing a little bit in advance.
Yeah, all that year.
I know they are busy in having dinner together. And, okay.
According to what you're saying, do you think that it's right to state what optimal microbiome and unhealthy microbiome is? Since every organism is different, even in human case. Yeah, we have some organisms that are defined healthy, but they are not known case. So in your opinion, is it right to define an optimal one?
I think it could be useful because it could be a way also to detect the human impact. But how to define is not easy, because of course there are a lot of things that can be discussed. For instance, we know that how can we define a fish in our seas that is not impacted for anything? Maybe in a marine protected area. Yeah, so I mean, it's there's a conceptual, and I think the solution is that the more data we collect from the different parts of our seas, and then maybe we can use big data to understand the core of taxa that can be useful. I mean, the same has been done for human, but it has been putting together data from many works, and I think here the problem that we have is the paucity of data, so there's still a few data, and if we could at least in theory define an optimal microbiota, that could be a reference. That could be a reference, but how we can achieve that's a discussion with fish biologists. Ideally, could be useful to detect how deviation of a microbiome or dysbiosis, for instance, could be also early warning indicators, or pollution, or climate change, or whatever. Or in aquaculture, for instance. In aquaculture, you don't have early warning, so you typically have the disease and then you have to treat it. But if you could have something that could give you an early warning, that could be useful.
So even if in aquaculture you are deciding what the fish is eating, there is the possibility to work that something is naturally
Yes. Yeah, I mean, in aquaculture, you are feeding with something that is artificial, is not natural, it's feeds, and so
The environment is not natural.
the environment is not natural. So the question is, the microbiome of these farmed animals is healthy, is optimal, is artificial?
the kind of sense that you are associated with optimal is something useful also for why, but it's part of the
Yeah.
We assume that wine is the most natural.
Yeah, maybe natural is better term than optimal. Based on the assumption that natural and pristine is optimal. But, yeah. It's not easy. But yeah, it's the same for chicken and poultry also as well. The microbiota is completely different, and the meat of farmed sea breed is completely different from the wild. If you have tasted, it's different. So is the feed, of course, is the microbiota, is what? Yeah, my feeling is that aquaculture will be funded a lot in the next years also as an opportunity for you, and yeah, compared to fishery, maybe where fishery is receiving a lot of subsidies to the sectors, and that are structural for the sector, but we know that the fishery fleets is decreasing, and I see attention by the EU on fishery in terms of maybe more strict regulations, in terms of technologies, greening, reducing the footprint and whatever, which is okay. But for aquaculture, I think that research and development, there's plenty to do, and there's a lot of research, of course, not only for the microbiome, but for everything. And especially aquaculture has grown in Asia, for instance, very low technology, a lot of pollution, very low quality of the fish. We are eating a lot, but there will be maybe a change also in our perception. So I don't eat shrimp from Asia very favorably, because I know how they are aquaculture. So if this is my perception, if we will have better food, higher quality, higher safety, and this will depend on the research. It will depend on the research, and this is why the EU. Last week I was talking at Stockholm, digitally, but it was you know that Sweden has now the semester in the EU, so it's the EU semester, which is held by Sweden, and the Parliament was organizing a very interesting interparliamentary conference on the blue circular bioeconomy. So it's not only blue economy, it's blue bioeconomy, so it's fisheries and aquaculture and algae and everything that is related with bio. That has to become circular and sustainable and so on. And what was emerging is that we need a lot of research. And especially looking at the Italian research enterprises, they are innovating not that much. Then we have a lot of bureaucracy. So opening a company of aquaculture is not easy in Italy, because you have to go through a lot of permissions, and you know that in the seas there are many institutions and government that have the power to decide, so you have to ask for permission to everyone, so it's not easy. And this is also part of the aquaculture crisis in Italy, because we know that sea bream market is dominated by Greece, is dominated by Turkey, is dominated by Croatia, and many enterprises has closed. So also to foster the Italian aquaculture, we have to foster innovation. So if we want to breed, if we want to farm sea bream, it will be difficult, because the products coming from Turkey, coming from Greece, they have less taxes, manpower, and so on. So it is difficult to become competitive for the market. But if you think at new aquaculture, new species, multi-trophic aquaculture, new target species, and so on, that would be an opportunity also in terms of companies, of works, of jobs, of research. And seaweed, also seaweed will grow. Corals, I don't know. Okay, sorry for boring you.
How trustable are the metabolic fat rates that you can reconstruct at the end of the metagenomic studies, for example?
Yeah, I think there the real challenge is to separate the microbial gene from the host, which is so there's a technical challenge. Yeah. And then I think if you overcome that problems, it's quite well established, the software that you can use now to reconstruct they're quite good. Yeah, but for the holobionts, that's a big challenge, how to separate, because you will get all the gene from the fish. I think that you have salt in circles. You have, no, not yet. That's a technological challenge, yeah.
But for a given metabolic pathway, you can just say release or there's no presence absent, or even a quantitative. You can quantify that metabolic pathway, or just giving presence or absence?
With metagenomics, no. I mean, it's just a number of copies. But maybe if you do metatranscriptomics, you can also evaluate the transcripts. But yeah, I'm not particularly expert in this, so I don't know.
I have a real question. What's the value in this field that is evolving so fast? Pioneering something, or just wait a few years and then apply successfully?
You mean in general?
Yes, probably. I perceive that I'm not an expert, but talking about something on the edge of but to say that just 15 years ago, many of the things that are now something like protein array were rocket science.
Yeah.
So it would be difficult to pioneer something that you know that in a few years already old or really destined to go take. So you as an expert in this field, how do you perceive it? You have the value of pioneering.
I mean, what my suggestion, I mean, when I was your age, I was really taken by, technologies, and I was really fascinated by achieving the data with the technology. So think of the Americans, they have the money, the sequencing, and my suggestion is that always consider the technology as a tool. So you can make your hypothesis and then ask yourself, which is the tool? Maybe cultivation can be a tool to demonstrate your hypothesis. You don't need metatranscriptomics. And this is something, it's a good point, because now my feeling is that everyone is sequencing, everyone to publish, especially on fast journal, like the one that we have. We have many open access journal now that for them it's just the technology, the data, and a few line of text and an abstract, and go on. But don't forget to make an hypothesis and to understand if cultivation or metatranscriptomics, which are the opposites, so 19th century and 2015, which is the best tool to demonstrate. That's the best advice I can give you. And we have to say that technology is giving a hand a lot. I mean, for microbes, just imagine that when I started, I spent months counting this. This is what I could do. Take samples and count. Okay, here I have 10 to the 9, here I have 10 to the 7, here I have 10 to the 6, here I have 10 to the 10. And that was all and then I did graphs and say, oh, that's more food, that's more pollutants and whatever. I really wanted to give a name to this. And this was not only fashion because it was curiosity. But then now we don't need to sequence everything. We have to use these tools to test our hypothesis. And then, but they can give a lot of, they can give a hand a lot. Thank you for the question, it was really interesting. Let's go.
Question?